All right, team, let's finish up our genetics, and we'll talk also about mutations, and then we'll move into the next unit, next topic, that is. Let's see. So last time we had just begun talking about this idea of polygenetic inheritance. Let me actually go back one slide. Okay, so what is polygenetic inheritance? As we talked about before, it's where you have more than one pair of chromosomes that are controlling the trait. So at this point, as far as the problems go, what we've seen is we've seen straight monohybrid and dihybrid cross, where you have a pair of chromosomes, right, controlling the trait. And then we saw multiple alleles where you still had a pair of chromosomes controlling the trait, but you had more than a pair of alleles, each individual only getting 2 of those. Well, now what I'm telling you is there can be multiple chromosomes with genes for the same thing. For example, human eye color. Another really good example that we can look at, let's do that really quick. Let me get rid of this screen. Sorry, I'm not being very proficient. Okay, doing my best. Here's another really good example. Let's say that we're at AB Tech. I wish we were. I'm tired of teaching on this computer. Let's say we're in class and somebody pulls the fire alarm and all of the people in the Sycamore building where we would normally be march out into the field in front of the culinary. And once everyone gets out there, they inform us, you know, it's not a fire. We're actually doing a genetics experiment. And what we want everyone to do is we want everyone to line up based upon your height. Find a line with other people that are your height and get in it. That's the idea. And so everybody starts moseying around, looking for people that are their height and getting in line, right? What if we wanted to graph that data? What would it look like? So if we make a graph of the data once we've done this, and on the y-axis is going to be the number of individuals that are a given height, right? Maybe in tens or whatever, because there might be quite a few people out there in the lawn if we did this. And then on the x-axis is height. Something like this. Now, when you're thinking about height, you know, what are the parameters? Well, what's the shortest someone could possibly be on campus? I know we do have, you know, we have a wide variety of height, right, on our campus. To be safe, we might start at 4 feet just to be safe. And what's the tallest anyone might be? Well, realistically, we could probably go up to 8 feet. There's probably not any 8-footers. I don't know if any human's ever been that tall, but you get the idea. So we have these parameters, the extremes at either end. If we graph this data then, what's the graph going to look like? It's going to be a nice bell curve, right? Something like this. Right? Which says what? Most people are average. Okay? But what I want you to understand is that we have now a continuum of phenotype. Right? We start out the shortest and we go through the average and we come back down to the tallest. And so anytime you see a continuum of phenotype, you assume polygenetic inheritance. And that is, in fact, what controls human height. Human height, how tall a human being is genetic. You have a genetic maximum for your height. Now, that does not necessarily mean that all humans reach their potential, right? And so you could imagine some things that could happen. Primarily nutritional deficiencies can cause an individual not to reach their genetic height, okay? But otherwise, you know, you have the potential. The point here being, it's not either short people or tall people, it's short people and tall people and everything in between, a continuum of the physical expression of a gene that indicates polygenetic inheritance. Okay. So that explains it a little further. Now let's go back to this PowerPoint here and let's look at an actual example of a problem that we might do that deals with polygenetic inheritance, okay? There's a lot of words in this problem, as you can see, but these end up being actually quite simple in terms of problems. No big deal, we can explain this. You understand what a zucchini squash is. It's a kind of a squash, okay? And it says that length is controlled by polygenetic inheritance, okay? And so these would be better as subscripts. And again, I apologize, but if you could just make a note, it would really be better to write it like this. So one variety, which is big S1, big S1, big S2, big S2, it tells you that variety is homozygous dominant. It has 25-inch fruits. And then we have another variety that's recessive. And we'll talk about what the ones and the twos represent in a bit. Okay. This individual that's homozygous recessive has 5-inch fruits. Now here's the thing, how many, you know, the S is just representing the length gene. I don't know why I chose to use an S, maybe for squash, it doesn't really matter. How many chromosomes must be controlling the trait? 4, right? So what that means is there's a pair of chromosomes with the length gene, and then there's some other pair further down the line also with the length gene. And what that means is each one of these chromosomes is essentially contributing 25% to the length of the squash. The ones and the twos, we're actually using to keep track of homology. So you know these are a pair of chromosomes and these are a different pair. And that's what the ones and twos represent. Okay, so now it says determine the length of the following if each dominant allele increases length by 5 inches. And then I just give you sort of these random genotypes here, and I want to know what the phenotype is. I want to know how long these fruits are. Now, here's the thing that you have to understand. There are parameters in which you must work, okay? What is the longest that the squash can be? If it's homozygous dominant all the way across, all big S's, then it has 25-inch fruits. That's as long as it can be, okay? What's the shortest it can be? Well, it tells you in the problem, if it's all homozygous recessive, it only has 5-inch fruits. So let's write that out just to remind ourselves, okay? Within those parameters, you must work, and it says each dominant allele increases length by 5 inches. So the way to approach it is you look at this genotype here and you say, okay, if this individual was homozygous recessive, if it was homozygous recessive, how long would the fruits be? Well, if it was homozygous recessive, it would have 5-inch fruits. But it's not homozygous recessive. We have this dominant allele here. There's only one dominant allele. So we have to do what? We have to add 5. So 5 is your base. You start at 5. And because we have one dominant allele, we add 5. So 5 plus 5 equals 10. So this individual will make fruits that have 10, will make squash that are 10 inches. What about this one? Well, same thing. If it was homozygous recessive, it would be 5. That's as short as it can be. Then we have to add 5 for each dominant allele. So is that a dominant allele? It is. So 10, 15, nothing there, nothing there. So this one's going to have 15-inch fruits. This one ends up being the same as the first one. What about this one? Start at 5, 10, 15, 20. Okay, so these are relatively easy problems. I'll show you kind of a little way to cheat a little bit. That maybe if you didn't see it right away, it's not surprising. I sometimes have to go over it a few times with students. So check this out. What if we did it like this? What if we said, okay, I'm almost out of room here. So what's the tallest it can be? Or what's the longest? 25. What's the shortest? 5. For each dominant allele, you just add 5. So you start at 5, and there's one dominant allele is 10, two dominant alleles is 15, three dominant alleles is 20, and then we're at 25. So when I give you one of these problems, obviously the math has to work out. I have to make sure I've worked out the math, or the problem won't make any sense. Okay. Another thing to note, check this out. What about this one? 5, start at 5, 10, 15. What about this one? Start at 5, 10, 15. It doesn't matter where the S's, the big S's are, it just matters if they're there or not, okay? So these two would have the same length. This one is heterozygous at both positions. This one's a homozygous dominant at one and a homozygous recessive at the other, okay? So these are relatively easy problems. You could expect to see one of these problems on the next test. Okay. All right, team. Believe it or not, that takes care of the genetics PowerPoint, with the exception of talking about mutations. So now let's have a few minutes to talk about mutations and what they are and what the implications are for them. And we'll see. We'll talk about some examples, talk about some causes, okay? So by definition, a mutation is nothing more than a change in the DNA of the organism, okay? Any change. It can be a big change, it can be a little change, it doesn't matter, okay? So if the DNA changes somehow, either it changes within the lifetime of an individual, or it could change from parents to offspring. You know, there could be a mutation during meiosis, for example. Either way, it's a mutation, okay? And as far as how often this happens, in eukaryotic cells, it's 1 in a million. Isn't that interesting? So when does it happen? Well, remember the replication process, the S phase of the cell cycle. Well, that's where the DNA is replicated or synthesized. And sometimes an error can occur during the replication process, and then you could end up with a mutation that way. Or it could just be that a cell mutates kind of on its own during its lifetime. The DNA just somehow changes. So oh, that should be an A for effects. My bad. There we go. Relatively large mutations are called chromosomal anomalies, and these affect part of a chromosome or possibly even an entire one. And we'll look at some different examples of that. And then we have point mutations, which now we're on the molecular level rather than the chromosome level, and it affects one or more of the base pairs. Remember those A's and T's? Oops, didn't do that right. A's and T's and T's and C's and G's. Well, perhaps something happens just to one base pair. Maybe that base pair gets damaged and it's gone. Or it could be a couple of base pairs. We'll talk about examples of that. We'll actually talk more about that when we do protein synthesis, because we'll then see the implications of this. Okay. So what are some causes of mutations? Well, they can simply be genetic. You can just inherit a genetic mutation from your parents. Really good example of a genetic mutation is the BRCA gene, which is a gene which predisposes women to breast cancer and men to prostate cancer. If you have two copies of the BRCA gene, you got one from your mother and one from your father, you have about an 80% chance of developing breast cancer throughout your life. And it also increases a male's chance of developing prostate cancer. So this is an example of a genetic mutation that you inherit that is clearly a negative mutation. Okay, but we have to be careful about this idea of mutation being negative because it's not always negative. Sometimes genetic mutations are positive. For example, there was a genetic mutation which occurred, we believe, about 40,000 years ago in humans, and that genetic mutation protected some of your ancestors from the bubonic plague, which swept through Europe, right? And so if these individuals had this genetic mutation, they were missing some proteins on the surface of their blood cells, and the plague was unable to infect them. And so that's an example of a mutation that's actually a beneficial mutation. And so don't just assume that a mutation is negative just because it's a mutation. It could have no effect on the organism that's obvious. That's probably the most common, or it could certainly be a positive mutation, as in the case of this mutation which protected some earlier folks from the bubonic plague. Now, here's the crazy thing. About 1% of the human population has this mutation that protects them from the bubonic plague today. Of course, bubonic plague is not a big risk, but the same mutation also protects these individuals from HIV, which is a virus which ultimately causes AIDS or can cause AIDS and can kill you. So although the plague is a bacteria and HIV is a virus, the same mutation protects these individuals' ancestors that are alive today from HIV. So a very good example of a good mutation. What else can cause mutations? We know that certain chemicals, chemical exposure, dry cleaning chemicals have been notoriously known as mutagens. We have seen this in people that work in that industry. They have much higher levels of certain types of cancer. I think pancreatic cancer particularly. And we believe that certain chemicals just denature the DNA. They change it. Radiation, right? UV, X-rays, etc. Right? This ionizing radiation can mutate the DNA. Drugs, including pharmaceutical drugs. And so just because you get a prescription for a pharmaceutical drug doesn't necessarily mean that it doesn't have risks of causing genetic mutation. And when you get the drug, you get a little pamphlet of information with it. And if you look closely, it will tell you, well, we did these studies on rats or mice or monkeys or some mammal. And we found that, at this dose, it caused potential mutations or something like that. So you can look at the rate of mutagenesis, heterogenesis, and carcinogenesis in your medication. Now, I'm not telling you that you might not still need to take the medication, because if it's a medication that you need, it's a medication that you need, but it does potentially could have inherent risks. Okay, so there's a little basic info about mutation. Now let's look at some examples of chromosomal anomalies, which are fascinating. And let me get rid of this and get back to my whiteboard screen. Okay. So, chromosomal anomaly. again, now we're talking about on the chromosome level, some major change. One of the most common chromosomal anomalies is trisomy or monosomy. And so now what's happening at some location, there is either in trisomy an extra chromosome or in monosomy missing a chromosome. Okay. What I mean by some location, remember humans have 46 chromosomes, 2 sets of 23. And so one of the most common trisomy events is trisomy 13, which you know as Down's syndrome. Okay, so the clinical name for trisomy 13, 13 being the 13th chromosome. There's 3 instead of 2. Now you might think, well, what's the big deal about just having an extra chromosome? Well, if you've ever known anyone or maybe someone in your family has Down syndrome, you know it can be a big deal. There are varying degrees of severity, but in its most severe form, it causes both facultative and physical handicaps in the individual. Okay, so be aware that these things happen. Monosomy is where you're missing one, and this is also can also be a big deal. How does this happen? Well, interestingly, it seems that it's the egg at fault here and not the sperm. And so remember, in meiosis, these chromosomes normally get pulled apart, but what can happen is when they line up, for some reason, the egg doesn't separate, and it ends up, one of the eggs ends up with both of the chromosomes instead of one, and then along comes a sperm with 1, and you get 3. Okay, so why the egg? That's the big question. And how do we know it's the egg? Well, we know it's the egg primarily because we have done lots of studies in this condition of Down syndrome. And here's an example. If you compare the age of the mother versus incidence of Down syndrome, if here's age, and we have to start at 14, don't have a baby at 14, y'all, but you could potentially. When we go all the way up to 60, you know, I mean, that's pushing it. And then this is the incidence of the condition of Down syndrome. It looks like this. Whoop. So exponentially more likely for, say, a 45-year-old woman to have a Down syndrome child than a 14-year-old. Chances of a 14-year-old having a Down syndrome child, 1 in a million. Chances of a 45-year-old is closer to 1 in 100. So it dramatically exponentially increases your chances if you wait until you're in your mid-40s to conceive a child. Now, why is this? What's the explanation? Well, unlike sperm, which are constantly being reabsorbed and regenerated, they're not more than a few days old. The eggs, or at least the precursor cells to the eggs that a woman sheds during her ovarian cycle, are there at birth. And so all of the things that the mother has been exposed to throughout her life, just life in general, radiation, chemicals, drugs, all of that stuff can take its toll on these eggs and ultimately cause this process where they don't actually pull apart very well in meiosis for whatever reason. Okay. So what's the take-home message? Well, you know, you don't want to have a kid when you're 14, probably. You're incapable of taking care of one. But if you want to have children, maybe plan ahead and don't wait until you're in your mid-40s. Okay. There are other chromosomal anomalies, of course. And let's get rid of all this for now. Another example of a trisomy event is very interesting, and it's trisomy 23. Now, why would this be interesting? Well, what's special about the 23rd chromosome is it is the sex chromosome, right? And so that means that this controls the sex or determines the sex of the offspring. So if you have a trisomy event, you can have a situation where you can have 3 X's or 2 X's and a Y. And so this is an interesting situation here, being that it's the sex chromosome. Remember, normal female karyotype is XX, and normal male karyotype is XY. So what does this all mean? Well, in this case, this has often been termed super female, and you'll see this still used. And these individuals have 3 X's. They present typically, at least physically, as females, which I guess you would expect, being they have 3 X's. But if you start measuring them, you quickly see that they're not quite normal in terms of their physical attributes as compared to what would be considered the normal karyotype. They tend to have larger hands and feet and longer faces. But suffice it to say, if there was a room full of people, you may not notice this. They would likely present as female in the eyes of most people. Okay, these individuals are generally sterile, although they can, with the hormonal treatment, sometimes conceive offspring. Okay, this one is interesting. I think the most appropriate term to use here is intersexed. And this term can apply to lots of different conditions. This is just one example of intersexed. It has been called Klinefelter's syndrome in the past. It's also been called hermaphrodism, which is considered an inappropriate thing to call it. Okay. What's going on here? Well, they have 2 X's as a female, but a Y as a male. And so this, presents some interesting issues to these individuals. Now, normally when an intersexed child is born, they typically have somewhat ambiguous genitalia. So they might have a smaller than normal penis and also a vaginal opening. Or their genitalia can be more like one or more like the other of the genders, okay? The typical scenario when an trisomy 23 XXY is born, the doctors almost always kind of pressure the parents or suggest to the parents, we need to do surgery immediately. The sooner we do surgery, the better, okay? And so basically they're saying, we want you to choose the sex of your child. Do you want to raise your child as male or a female? And they are almost always pushed into the female by the doctors. The reason for this is because the surgery is easier. As they say in the industry, it's easier to make a hole than a pole. So it's easier to lop off the penis and make a vaginal opening than it is to close the vaginal opening and make a penis. It's just the logistics of it, okay? But think about the implications of this for this person. Right? You are choosing the sex of your child at birth. When these individuals begin to grow up, and certainly when they hit puberty, they begin to feel more like one or the other of the sexes. And what if you choose the wrong sex? How horrible is that? And so there's a faction of individuals to which this was done at birth that say, don't do this to people. Wait until they are old enough to decide for themselves, right? But this also, unfortunately, creates issues because we live in a binary society where you're either supposed to be a male or a female. And so, questions arise like, what bathroom do these individuals use? What gym class do they go to? Stuff like that. So it can be very complicated. And I just want to tell you, until we accept that non-binary is a thing, and that this is just one example of an individual that's intersexed, and that all sorts of other things can happen in utero that can cause an individual that is genetically female or genetically male to certainly have features of the other sex and certainly mentally or psychologically identify with the other sex. Okay, so we're starting to get it, I think, but we have a long way to go in realizing that we live in a non-binary world, or at least we should. Okay. Some other things that can occur is, now those were dealing with entire chromosomes. Here's a wacky thing that can happen. Remember crossing over, which occurs in prophase 1 of meiosis? I'm going to do 2 different colored chromosomes here. And crossing over occurs, and remember, these things switch pieces of chromosome, which is what you would expect. So this piece breaks off and attaches here, and this piece breaks off and attaches here. One really bizarre thing that can happen is called translocation, where for whatever reason, this piece doesn't break off, and this piece does, and it ends up over here. Now think about the fate of these. We're in prophase 1 of meiosis, right? These get pulled apart, these chromosomes get pulled apart. This one ends up in one, let's say it's a sperm, and this one ends up in another sperm. If this sperm gets the egg, you got this extra piece of genetic material. If this sperm gets the egg, you're missing a piece. Either way, it's an issue, okay? So that's one thing that can happen. Another weird oddball thing that can happen is this. This one always I find interesting. And you'll find it interesting too, I hope. It's called inversion. This piece breaks off and it attaches to the top of the same chromosome. Okay. Now this piece might go ahead and end up down here, like so, like it was supposed to. But this piece didn't end up over here. And so now you got a similar thing. This is called inversion. And you know, the genes don't line up anymore. First of all, these 2 chromosomes aren't going to recognize themselves as being homologous. Maybe we'll just do this to keep it a little simpler. Let's just say that piece never broke off at all. Okay. So now, you know, the genes don't line up. You might look at this and think, well, there's no big deal. It still has all of its genome, but the order of the genes is shuffled. And so this could potentially create a problem down the line when meiosis occurs and these are pulled apart. They're not going to recognize each other. They may not pull apart properly, and it could lead to a trisomy event later. Okay, so these are just some weird things that can happen. Translocation and inversion. Now, I think we're going to stop there. And, you know, what you have to realize about mutation is you can't control it, right? You don't have any control over mutations. Mutations just occur. And so because of that, we have to realize that, you know, we can try to protect ourselves from mutations, but even in the best case scenario, they're still going to occur. Okay. So this ends the discussion on genetics, and there will be a video with some of these sheets that we talked about. And let me just show you where those are again, just so you remember. We have these sheets that say more genetics problems, still even more genetics problems. At this point, you should be able to work any of those problems, and I would suggest you start doing that. Start working these. I'm going to pick the best of the 3, and I'm going to make a video of me actually doing the problems and make sure everybody understands. There's not gonna be a video for all 3 sheets, y'all. So you just have to, from what we've done so far and from the 1 sheet that I do, you should be able to answer the other questions. Okay. All right, I'm gonna sign off for now. I hope you have a great rest of your day, and I'll see you soon. Bye.